Am I audible? And can you see my screen? Can you? Is my screen visible? Yes. Okay. Sure. Thank you. Okay. Hi, welcome to the problem solving session 5 for Biochemistry NPTEL course. And today's session will cover lipids. So today's session is more of a theoretical session because it's the topic of lipids and exploration of the topic of lipids. But before I start, as I mentioned in the previous session, these sessions are meant for your doubts and your clearance. So until now, if you have any doubt for any topic which is not clear, let me know. I will put put slides for those in the next session. Any topic done so far. Next week, if I'm correct, we will be doing bioenergetics and glycolysis. So those parts I will cover in the next session. But if you have any doubt for any of the topic from for the first 5 weeks, please let me know. You can put that in the chat box, and I will also pin that message so that if people can see if they join later. And I can work on those in the next session because the motive of these sessions are for your problem solving or your doubt clearance. So if there are any such topics, please let me know, or if there is any topic that you want more of numericals to do, so I can put more slides on those so that you have a good practice on the numericals as well. Okay. So yeah, so for that, so do let me know if you have any such topics that you need more help on. So let's begin today's session. So like I said, today is more of a theoretical session. So if you would have seen the week's lecture notes and the sessions, they're mostly about lipids, kind of lipids that you find and the different types of lipids that are there. What are their roles? Where are they found? And then we had a very small portion on catabolism and anabolism. And most of the questions and most of the topics are about lipid derivatives, lipids, what are their biosynthetic pathways, and all that. So as you can see, they are all theoretical. So I have put them in the slide according to how it was in the lecture notes. And we do have several questions. So let's try to attempt those questions and understand the topic better. Right. So before we begin that, so lipids, what are lipids? Lipids are heterogeneous group of naturally occurring compounds. These compounds, if you see all the lipids that you consider, if you look at these compounds are related more by their physical nature than by their chemical properties. These are insoluble in water and are likely be so because they have more of a hydrophobic, like hydrophobic groups. And that's why, because they're insoluble in water, that means that they don't have hydrophilic groups. So for them to be soluble in water, these lipids have to have a hydrophilic component that will enable them to be soluble in water. By itself, lipids is insoluble in water, but because it has a hydrophobic group, it is more soluble in non-polar solvents such as ether and chloroform. Right? This is very important when you are looking into lipids. We will have a broader classification in the coming slides. So there are two types of lipids. We have storage lipids and we have membrane lipids. The membrane lipids are polar, and if they are supposed to be polar, they have to have a group that is hydrophilic in nature. So you will find these phosphate groups here, or you will find the alcohol groups, or you will find these saccharide groups or sugar, which enables them to be soluble in water. So for membrane lipids, because they are also facing, you can see the image in top. If they are membrane lipids, they have to face the aqueous environment outside the cell. So they have to be polar in nature so that they are able to build that boundary. So they need to be polar. Whereas storage lipids, which are the fats that we call, they are of the insoluble in water nature, and they are mainly the triacylglycerols or TAGs. What you need to remember in lipids specifically are their structure, basically what is the backbone, what number of fatty acid chains that they have, and what is their classification. We'll do so many questions today that it should give you a good practice till the end. And we'll all do all these different names in the coming slide one by one. So let's begin with just a simple classification of lipids. So we have one as simple lipids and one is complex lipids. What are simple lipids? Simple lipids are basically esters of fatty acids. So these fatty acids, when they are connected to the backbone, they are bound by the ester linkages. So these esters of fatty acid with various alcohols, these are simple fatty acids or simple lipids. If you want to compare this right now with complex, complex means esters of fatty acids containing groups in addition to alcohol and fatty acid. So it's not just fatty acid and alcohol. You will have one additional group. I told you already that, for example, phospholipids will have a phosphate group, glycolipids will have a glycate group. If it's only fatty acid and alcohol, then it is simple lipid. Examples of simple lipids, which is just with fatty acids and alcohols, are fats and waxes. Fats are the TAGs that we just saw. TAGs are triacylglycerols. And then you have waxes, which are basically esters of fatty acids, which are the fatty acids have high molecular weight. Okay. Whereas in complex lipids, you have phospholipids and you have glycolipids. Phospholipids, as the name suggests, they will have a phosphate group in addition to fatty acids and alcohol. And glycolipids, they have fatty acids, sphingosine, and carbohydrate. So we will see their example and we will see their structure as well. Okay, so here on your slides are simple lipids. On the left, you have waxes. As we have already seen, they are long chained carboxylic acid groups. So those form the fatty acid part and then they are connected to the alcohol. And you can see they mostly are these big cetyl palmitate. So you know palmitic acid. And then you have these ester formation which make them wax or similar kind of long chain carboxylic acids. Whereas in fat, you see they will have this glycerol backbone and then they have the three fatty acids. So tri-acyl group and glycerol. So tri-acyl glycerol. Okay. Then you have here single fatty acid. So single fatty acid. This is called mono unsaturated fatty acid. This is polyunsaturated fatty. Unsaturated means double bonds. Unsaturated means double bonds. Saturated means single bonds. And this is what we mean. So fatty acid that you see here or here or here, if you look at the fatty acid composition, this is how fatty acid looks like. The fatty acids are basically carboxylic acid. Carboxylic acid is COOH, and then there's CH2, CH3, CH2 groups. So they are carboxylic acid with long aliphatic chains. These can be branched or unbranched, right? The fatty acids that occur naturally possess carbon numbers in even numbers. So C16, C18, C20. You will know these names as oleic acid, palmitic acid, stearic acid. All these are um, carbon in even numbers. We'll see that in a while. And these fatty acids therefore become major components of lipids, and we will see how they are forming phospholipids, triglycerides, and cholesterol esters, right? So look at the first one. It says it is saturated. Saturated means, remember, saturated means it's a single bond. All the carbon are connected or are joined to each other by single bonds. And this is palmitic acid. So can you tell me the total number of carbon here? Can you count and tell me how many carbon does this amino acid or this fatty acid has? You could type in the chat box how many total number of carbon does palmitic acid has. So you basically, so what you can also do is you can simply count the number of C's here on the structure. On the structure also you can count. But this is the way how you write the fatty acid when you write in general formula. So you write CH3 for the first C group, you write COH for the end, and whatever in between, it we know it's CH2, and we just tell number of, so CH2 bracket N. So how do we calculate? C14, so 14 times CH2, so C14 and one here and one here. So 14 plus 2, 16. So palmitic acid has 16 carbon. Similarly, stearic acid, 16, 1 plus 1, 2. So 18 carbon. So these are both saturated fatty acid. While when you say unsaturated fatty acid, you mean that there will be a double bond. So here you can see there are two double bonds. So when there is a single double bond, it's called mono unsaturated. When there is multiple double bond, that's called polyunsaturated fatty acids, and that's what we saw here. When they had multiple double bond, that was called polyunsaturated fatty acid. When it's a single double bond, it's called monounsaturated fatty acid, right? Okay. And this one, if you see here, if you still count the number of C, it will give you the total number of carbon here. So you can look into this. 1 plus 5, 6, 7, 8, 9, 10, 10 plus 7, 17 and 18. So 18 is linoleic acid unsaturated. So you can count the number of total number of carbon. That also tells you what is the fatty acid we are referring to. So you can remember the fatty acids based on their classification, which is determined by number of carbons, their length, and the saturation, right? So this is what I was referring to as saturated fatty acid and unsaturated. Saturated means single bonds. There is no double bond between carbon. And because they are saturated, they have very high melting point because they are single bonds. So they're very tightly packed with each other. And that's why they're also straight rod-like shaped. But unsaturated, which has this double bond, which basically creates a structural kink, and this disables them to group their molecules in the straight. So there's a kink, so they will bend, so they don't have a straight structure, right? And they have lower melting point than saturated phase. That's why the name saturated. Saturated means it takes a longer time for it to open up, while unsaturated open up quickly, right? Okay, so this was about simple lipids, which is just this fatty acid chains that you saw. So you saw the fatty acids, right? Here you had the COOH group. So this COOH group on its C terminal is interacting with the glycerol backbone OH, and that gives rise to the ester bonds, which is where these get connected. So this forms the acyl, so tri-acyl groups and the glycerol, right? Very simple chemical reaction. If you remember the chemical reaction, you remember the name very easily. Okay, now this was simple lipids. Let's do complex lipids. So complex lipids, let's begin with phospholipids. Now, like the name suggests, phospholipids will have phosphoric acid, will also have a nitrogen base, and then an alcohol, and then fatty acids. Okay. And these lipids generally are found in the cell membrane because they have to make it polar in nature, and that provides a fluidity to the membrane. If you remember uh, that the cell membrane are fluid in nature, and that's because of these polar groups on the membrane. So now if you see, there are two types of phospholipids. Do you know the two types of phospholipids which are there? The structure is already given. The name of the phospholipid is also there. Just give me the broad classification names. Can you guess it from the backbone that is there? Sphingophospholipid, that's for one, and the other one? Glycerophospholipid. Excellent. So, like someone has mentioned, it's glycerophospholipids and sphingophospholipid. This is also there in your notes. So as you can see, phospholipids themselves further are classified based on what kind of group they're attached with. So if their backbone is glycerol, that's glycerophospholipids. If the backbone is sphingosine, it's sphingospholipids. So the name comes from the backbone. So if glycero, it means the backbone is glycerol, and because it's a phospholipid, it has fatty acids and a phosphate and an amino alcohol. Similar to this, in sphingosine, the backbone is sphingosine. That's why the name is sphingophospholipid. And that's giving you the alcohol here. So the group, for phosphate linkages and the fatty acid. So what you will definitely have between these two groups is phosphate and fatty acid and an alcohol group. The backbone changes and that gives a name to the backbone. Now why it is not simple? Because it is not just fatty acid and glycerol. If that had been the case, this would have been a triacyl glycerol. But here you have a phosphate group. Here also you have a phosphate group that makes it a complex lipid. So this is just first complex phospholipid, which is so first complex lipid, which is phospholipid. The next kind here, which is what we will now look into, the first classification here, which is glycerophospholipids. So glycerophospholipids are also called phosphoglycerides. These are again membrane lipids. Here, as we have already seen, you have two fatty acids which are attached by ester linkages to the first and the second carbon. What happens to the third? In glycerophospholipids as well, you have further classification based on the group, which gives the amino alcohol here, right? And so the basic one where you have just an H, making it an not no complicated group, just an H, is the phosphatidic acid. Every other glycerophospholipids is a derivative of a phosphatidic acid. This you should remember. These kind of words, what are the simple component, what are the derivatives of each of these complex lipids, what are the groups on those complex lipids, where are those present? These are few things that you should remember in lipids. Structure, once you crack it, that what does the structure actually compose of, it should be easy for you to understand the structure. But what you need to remember is where they are present and what are they derived from. So here, glycerophospholipids, whichever classification you see, they are all derivatives of phosphatidic acid. Why is that? Because phosphatidic acid is the basic. If you have just H, it's phosphatidic. The moment you change your X, which is the alcohol group here, be it ethanolamine or choline or serine or glycerol, the names is easy to understand. It's phosphatidyl because phosphatidic acid. So phosphatidyl, whatever is the group here, ethanolamine, choline, serine, whatever the group is here. But all these glycerophospholipids, first of all, they are complex lipid and they are derivatives of phosphatidic acid, right? So this was glycerophospholipids. What about the other one, sphingophospholipids? These are derived from ceramides. Now, sphingosine, one fatty acid, and the second one is where you have the alcohol, right? So you have sphingolipids. These are composed of one molecule of long chain amino alcohol or of its derivatives. So you have sphingosine or derivatives, one molecule of long fatty chain, and the polar head that is joined by a glycosidic linkages, generally a phosphodiester. So that's why it's phospholipid, but it can also have some other structures, which you will see in the classification. Why it can also have another clue. But let's say your X here is ceramide. It has H, that's the ceramide. The basic one is called ceramide. The X, as it keeps changing, if it becomes sugar, it becomes neutral glycolipids. So if it has others, so mostly you will see that it has a glycosidic linkages. It means it has a sugar, or it can also have a phosphocholine, which gives it the names of phospholipids. So if it has a phospholipid, it becomes sphingomyelin. So this one is a sphingomyelin. But if you keep changing it to a sugar group, this base first of all becomes neutral because it is no longer soluble in water, and then it has different names. Okay. Well, this was just first of the complex lipid. Let me go back to that slide again. So complex lipid, first was phospholipid, second is glycolipid. Glyco is the name for sugar, and this is very simple to remember, so this is not very complicated. So you have complex lipid, which is glycolipid. Okay. Now in glycolipid, the name suggests it has to have a sugar. So any compound which contains one or more monosaccharide residues which are connected by glycosidic linkages, that's called glycolipid. Right? And they are mostly found in brain, nerve cells, and tissues. If I say the name, you will know it just by the name of it. So if you can have cerebrocytes and gangliocytes, the name says it. Cerebral means brain. So these kind of lipids are found in brain. Ganglion means ganglions, which are found in neurons. So these kind of lipids are found in neurons. Which of these? These are glycolipids. Now see these structures here. They are both sphingosine. Let's look at the previous slide. Sphingosine here. fatty acid here, and an X group here. If this X group is an H, it's ceramide. Okay. But if this X is a phosphocholine, it becomes phospholipids, so sphingophospholipids. But as when it becomes sugar, be it a single glucose sugar or a complex oligosaccharide, it becomes this glycolipid. If it is a single glucose, it is called cerebroside. It's found in the brain, and it is because remember it is not soluble in water. It acts as an insulator there in the brain. Similarly, in ganglion or neurons, you have gangliosides, which are composed of the sialic acid residues, and they provide the role for insulator. So why they are insulated? Because they are not soluble in water, and that's why it's very simple to remember. Right. So this is the broader classification. We've already done some of it, and the others are also following the similar patterns. So like I said, storage lipids are neutral. So you have TAGs, triacyl glycerols. Membrane lipids are phospholipids, glycerololipids, sphingolipids. We have already looked. Similarly, glycolipids can also be sphingolipids and galactolipids. So this three we have done, the first three. The fourth one here is galactolipids or sulfolipids. So as the name suggests, galacto. So first of all, it's glyco. So it has to have a sugar, and then it has a sulfur. So it is with a sulfate group. So it's in glycolipids. And then you have this archaebacterial ether lipid, which is only found in archaebacteria, where you have a glycerol backbone. Then you have diphthenyl, diphthenyl, 2-diphthenyl and phosphate, and again a glycerol backbone with a phosphate, which is again connected by glycerol. So this, as you can see, this is very rigid structure. And it makes sense why, right? Because they are found in archaebacteria. And where are archaebacteria found? Any idea? What are called archaebacteria? Which kingdom are we referring to here? Okay, maybe you can answer me in yes or no. So are these bacteria found in simpler, exactly, extremophile. So these archaebacteria, they're found in extremely harsh conditions. It's very high salinity, very high temperature, very high pH conditions. So what they need is a tougher membrane, a very resistant membrane. And that comes from the kind of lipid they have on their membrane, that is very strong, that is very tightly linked. So that's why they have these ether lipids, which are bigger and which are more resistant and stronger, while the others are comparatively simpler. So these are the broader classification. This is what you should remember: storage and membrane lipids and the different types of them, right? Okay. Then, so we are done with lipids, complex, simple, or, and the next type of lipids are sterols. The most common sterol that you would generally use in your daily use word is cholesterol. So cholesterol. So sterol is the backbone. So what is the backbone here? What is basically sterol? Sterol is basically a structural lipid. As you know, it's present mostly in eukaryotic cells. What is the characteristic structure of steroid? When do you know it's a steroid, or when do you know it's a sterol? It has 4 fused rings. 3 of those rings has 6 carbon and 1 has 5. So 3 groups of 6 carbons, so hexamer, and 1 with pentamer, so 5. These 4 will be linked to each other, and that's characteristic steroid nucleus. Nucleus means the backbone or whatever is connecting the other groups. So nucleus of steroid, and on that we have cholesterol, which has this polar head and a non-polar hydrocarbon body. So polar head and a non-polar. So polar head here and non-polar hydrocarbon. When this is the structure on the steroid nucleus, this is cholesterol. Okay, and this non-polar hydrocarbon body, this can be as long as 16 carbon fatty acid. Okay, and because it has both polar and a non-polar end, this is amphipathic in nature. Okay, all right, so this is cholesterol. Now similarly, you have vitamins. Now what is vitamin? Vitamin, again, a general class of compound, which is again fat soluble or water soluble. So you can see the classification. We all know vitamin complexes B, A, D, E, K. So what we are concerned here is with fat soluble. So we are more concerned with A, D, E, K. We also have aqueous or water soluble vitamins, which are B vitamin B complex and non-vitamin B complex, which is C. So B and C are water soluble. A, D, E, K is fat soluble. Okay. Now, fat soluble, as we know, have just seen, they are vitamin A, D, and E, K. You should remember they are all derived from these isoprene molecule. Every vitamin A, D, E, K structurally are isoprenoid compounds. They all have this basic uh, structure of an isoprene backbone, which is CH2 double bond C, which has the methyl CH3, and then you have a CH and again a CH2. So isoprene. Now, vitamins are basically these isoprenoid compounds by which are synthesized by condensation of multiple isoprene units. So you can see here they have this isoprene. So for vitamin D to form, the precursor is 7-dehydrocholesterol. And then under UV light, as we know, vitamin D is favored under sunlight, if you remember. So under UV light, you have the by two steps conversion of this into vitamin D3, which is cholecalciferol, right? And that can again get transformed into other kind of compounds. What are these different kind of compounds? You could read more in detail to know about the compound, but what you should definitely remember, and my advice to you is remember the precursor of your vitamin. Where does it come from? Which is the first molecule? Where does it start from? And how many pathways are there? And what is the name of the vitamin E? Sometimes the name of the vitamin tells you the pathway of the vitamin. For example, here you know it's cholecalciferol for vitamin D3. That's already calcium. It's a calciferol and it's chole, so it has to come from a cholesterol and it has a calciferol backbone like this. Similarly, vitamin A starts from beta carotene and that becomes retinol and then gets isomerized to form the retinal. So retinol and then retinal. So retinol and retinal is what refers to vitamin A. Similarly, you have vitamin E, vitamin K, which is a blood clotting factor, also called phyloquinone. And these are just vitamins that we know of. There is also warfarin, which is blood anticoagulant. You have ubiquinone, plastoquinone, dolicol. These are all synthesized from these similar kind of pathways. They're all used as either electron carriers or hormones, but they are all coming from these long hydrophobic group, which gives rise to this group of lipids, right? And that's why we are reading it here. And lastly, the last group of lipids that you need to know are the derived lipids. So these lipids, you know, the major classes of lipids, something which is derived from lipid by adding a group is basically a derived lipid. So these compounds are produced by hydrolysis of simpler or complex lipids. And because they are just, you know, like they can break away the complex or simple molecules of lipids. These generally involve the singleton units, so fatty acids, or glycerol, or steroids, or alcohol. These are derived lipids. So, for example, acyl glycerols, not triacyl. Acyl glycerol is just one. Triacyl glycerol is glycerol with three fatty acids. Just a glycerol with just one fatty acid is acyl glycerol. So, acyl glycerol or cholesterol or cholesterol esters, they are neutral in nature because they don't have any charge. And we already know how. And from so what falls under them are hormones, all the hormones that we know of, vitamins that we had just read, and we have ketone bodies, which are also derived from lipids. Right. So we have seen different classes of lipids and how their structure are formed and how they look like. So let's now take one step ahead. Let's try to solve questions and then try to understand more of these compounds. So here are 4 questions on the slide. Take your time and give me the answers, and we will discuss each of these answers one by one. The first question is the low molecular weight soluble lipids that can be transported in the blood plasma without the requirement of transporting protein is which are the following lipids. Then the molecule involved in the aggregation of platelets in the first stage of blood clotting is which one of this following? Next, Niemann-Pick, a lipid storage disease, is caused due to deficiency of which of the enzyme? And the last one, the immediate precursor for cholesterol is which of the following? Right. So please let me know whichever answer you feel like in the chat box, and then we can discuss. Okay. Okay. D beta galactosides A. Okay. You think. Okay. Wait for all the four answers and then. B last one. Okay. Okay. So B. Okay. I will. Let me first give the answers and then let first I'll wait for a few seconds for you to see if your answers match, and then I will give each answer. We'll discuss these of this question one by one. So all of these are already there in the lecture notes one by one or as a part of your topics. I did not take them up individually because doing it in a question makes more sense because you can compare and directly go to the theory behind it. From books and for aggregation of related. All right. Yeah. So I have given the answers, and if you got it right, then well done. If you didn't get it right, that's also fine. But the what was more important is you think in these lines. So we'll see what is happening to each question. So the first question is the low molecular weight soluble lipid that can be transported in blood plasma without the requirement of transporting protein. Basically, in this question, you have so many words that gives it away that the answer is ketone bodies. So what is the ketone bodies? It's basically these. Wait, let me give the pointer. So the acetone, acetoacetate, and butahydroxybutyrate. So these are the ketone bodies. Okay. So these ketone bodies, they are very small, as you can see, and they are water soluble. So the name was soluble lipids. So these are soluble because of the OH and the O group, and they are derived from fatty acid metabolism. And because they are so small and they are soluble, so our soluble is checked, our transported in blood plasma is checked, so they don't require the assistance of transporting protein. That's why the answer here is ketone bodies. Now looking into the contrast, triacyl glycerols, if you see the triacyl glycerol structure here, glycerol backbone and fatty acids, these are basically all hydrophobic, so they are not soluble. So the first point itself is cancelled in this. And for your information, uh, triacyl glycerols and the phospholipids, they require lipoproteins for transport. If you have read, there are chylomicrons, there are VLDL, HDL. All these help the transport of triacyl glycerols and the phospholipid. Similarly, sphingolipids They are also amphipathic, but they are transported as lipoprotein complexes. So for lipids to be transported, they need this conjugation of lipoproteins that help them transport in the blood. But ketone bodies are very, very small in structure, and that's why they don't need assistance of any further protein, right? Okay. Next was this one. This one says the molecule involved in the aggregation of platelets in the first stage of blood clotting is what? And we know this, I'm pretty sure everyone knows it already. It's thromboxane. And thromboxane again is the lipid derivative. So we have read about derived lipids. So this is derived lipids. And these are produced from arachidonic acid. And as we have read, we're doing vitamin, so vitamin K and thromboxane, they take part in the formation of the clots when we get a cut, right? Now, what are the others here? The chylomicrons here, as I have just told you, these are the lipoproteins that help in transport of fat, right? And then you have these two compounds called prostaglandins and leukotrienes. So prostaglandins, they are anticoagulant in nature, and leukotrienes are, you will see them taking part in inflammatory responses. So they are not involved in blood clotting. So that is also a cue for you to know which this is not the correct answer, right? Next, this one is enzyme. Sorry, this is a disease which is caused due to the deficiency of an enzyme. The enzyme is sphingomyelinase, right? Now, why is it a problem? So this enzyme, this breaks down sphingomyelin into ceramide into and phosphocholine. The basically, if you remember sphingomyelin, you have phosphocholine group and ceramide backbone. They just break the bond between these two. So you have a ceramide and a phosphocholine, right? But when this enzyme is not working properly or there is deficiency, this sphingomyelin gets accumulated in the lysosome. Okay. And especially in liver, brain, and bone marrow. And if because it is, if there's a over it gets an excess in amounts that has negative results that leads to cellular and organ damage. So that's why this is a disease. This is a problem. But the other enzymes, glycosidase, and the ceramidase, they also are deficiency of these enzymes also cause problem, but that's not Niemann-Pick. For galactosidase, you have gangliosidosis and Krabbe disease. Glycosidase Gaucher's disease, and ceramidase is Farber disease. So, like I said, these are mostly theoretical, so there is nothing to explain. These are things that you can remember. So you can use this slide whenever you are trying to revise. Right. Yeah. Coming to the next one, which is about the immediate precursor of cholesterol, and the answer was 7-dehydrocholesterol. So we will see that it is the immediate precursor of cholesterol in the cholesterol biosynthesis pathway, and it is basically so the 7-dehydrocholesterol is converted into cholesterol by dehydrocholesterol reductase, right? While the others, which are cholecalciferol and ergosterol and ergocalciferol, you will see that they are not part of the cholesterol synthesis pathway only. They are all of different pathways. So cholecalciferol that we have already looked into, it's a vitamin D3 component. It's not a precursor for cholesterol. Ergosterol is for sterol, which is for vitamin D2, and ergocalciferol is again vitamin D2, but it is a derivative of the ergosterol. So ergosterol, the sterol here is calciferol. But none of these are for cholesterol synthesis. Okay, right. Moving on to the next question. So there are set of 5 sentences for triacyl glycerols. Uh, you have to guess the correct ones. So TAGs are composed of 3 fatty acids, each in ester linkage with single glycerol. Second, the carbon atoms of fatty acid are less reduced than those of sugars. The oxygen of triacyl glycerol yield more than twice as much energy as carbohydrate oxidation. These are amphipathic molecules, and the primary role of triage is energy storage and metabolism. Let me know which ones are correct. ACE. Okay. Maybe just wait for a few more seconds. One else wants to answer. Okay, I see someone typing. Let the person type. Okay. Okay. So let me give the answers. Right. So, second, yeah. The first one is correct, as we all have already seen multiple times in the structure. B. Let's look at the first. Look at the correct answer. So the third is correct. That oxidation of triacyl glycerol yield more than twice as much energy as carbohydrate oxidation. And third one, fifth one, which is already we have seen classification. Let's look at the fourth one first, which is incorrect. Triacyl glycerols are amphipathic. Incorrect, because that's why we have told that it's not amphipathic. It's totally hydrophobic. That's why it's neutral. And lastly, carbon atoms of fatty acids are less reduced than those of sugar. It is wrong, because it is more reduced as it has less number. So your fatty acids are long hydrocarbon chains, right? So there are very, very few oxygen atom, if any. There's only in the carboxyl group. So less number of oxygen means they are more reduced. They have more hydrogen bonds. So they are more reduced than those of sugar, because in sugar you do have those oxygen molecules. So more the oxygen, more oxidized, more the hydrogen, more reduced. So carbohydrates have long chain of or fatty acids have long chain of carbohydrates, so which are mostly connected, which are all H bonds. So more H, more reduced. And that's why it says less reduced. That's why it's incorrect. Okay. Okay. This is very interesting. So you have a fatty acid composition of three profiles. The profiles are like it says, if it's darker blue, it has C16, C18 saturated. Then if it is middle blue, then it is C16, C16 unsaturated. And the lighter blue are C4 to C14 saturated. And then for these three compounds, so animal fat, groundnut oil, and butter, these are the three profiles you are looking into. So can you tell me which one this refers to, which one this refers to, and which one this refers to, based on the proportion of saturated and unsaturated fatty acid the profile shows? CBA. Okay. Anyone else wants to answer? Okay. All right. Just a second. Let me give you the answer. It's CAB. Okay. Let me also give you this. Did I get it right? C. I am not sure. I did say CAB. But well, the profile is here. Let's just, you can ignore the answer. The profile is here. So animal fat A, butter is C, and this is your groundnut oil. So BCA. The answer should be BCA, not CAB. I'm not sure why it went wrong. So this one is olive oil, groundnut oil. This one is butter, and this one is animal fat. Okay. So the answer should be C, A, and B. So BCA. Yeah, BCA, if someone got it right. Correct. I'm sorry for this mistake here. I'll correct it while uploading. Right. So what do we see, and where is this from for your reference? If you want to refer to this, this is in Leninger book. This question is from Leninger. And this profile is also there in the Leninger, so you can read up on that. So basically, it tells that if you look into the composition of lipid of olive oil or any groundnut oil, you will see that it mostly consists of this unsaturated fatty acids and a very few proportion of the saturated, which is again from the lower chains. The higher chain are comparably higher, but it's mostly unsaturated. And that's why you'll often hear oils as being called as polyunsaturated. So MUFAs and PUFAs, if you have seen ads of oils, then you will remember they keep saying MUFAs and PUFAs. So those are all polyunsaturated. So here you do see that the unsaturated fatty acid composition is very high. Now compare this to butter, which has comparatively equal proportion of the unsaturated and saturated long chain fatty acids, and then you have saturated fatty acids. But when you look at animal fat, you have mostly unsaturated fatty acid and saturated fatty acid in a similar composition with very less low weight uh, hydrocarbons. So this profile are characteristic of these different kind of compounds. Okay. Okay, the next set of questions. So the left one says which of the following lipids are component of chloroplast, and the right one says which of the following molecules are derived from cholesterol. Let me know your answers and then we can discuss. A. A. Okay. Second questions A and B. Okay, A and B. Okay. I'll wait for the other person to respond and then we can discuss. Okay, A and A. All right, let me first show you the answer. Okay, so the answer for the lipids found in chloroplasts are sulfolipids and galactolipids. And the answer for molecules derived from cholesterol are testosterone and cortisol. Let's take this question one by one. So the first one says, which of the following lipids are found in chloroplasts? So if you look into the structure of chloroplasts, you will see that these are the kind of lipids that are found in the thylakoid membranes, which are basically galactolipids and sulfolipids, which are again, so you can see these are the three glycolipids of chloroplast membrane. So you have MGDG, DGDG, which are galactolipids, and this you have sulfolipids, which is the third one, right? Now, what about the other two? Sorry. So you have the ether lipids and sphingolipids. Just by names, as we have already done it multiple times now. So ether lipids are found in so archaea bacteria or other specialized tissue, but not in chloroplast. And sphingolipids, we definitely know, are found in animal cell membranes, where also we know they're found in the brain or in the neurons, but they're not found in the chloroplast. So in the B option, you do the sphingolipids is incorrect. In the C, either is incorrect, and in D, both of them are incorrect. So that's why the answer is A, which you got right. So good job there. Now, next one is talking about which is derived from cholesterol, testosterone, and cortisol. If you look into their pathway, this is for testosterone. You see in the pathway, you will find a cholesterol. Similarly here, when you have cortisol, you start with cholesterol. And you also see something called convergence. So you have cholesterol giving pregnenolone. It is also here. This one component can give rise to three different, like this, you can give rise to adrenal androgens, and it can give rise to progesterone, while it can also at the same time give rise to 17 hydroxypregnolone, which gets converted to DHEA, which is again here. So all of these are converged, all of these are connected, all the pathways converge somewhere or the other. And why I'm talking about convergence and divergence, it will become clear to the second half of our lecture today, right? But for the time being, remember, uh, a pathway, a series of pathway can give rise to different components. Here, testosterone and cortisol both are derived from cholesterol, while the other three, which are your dolicol, retinol, and your vitamin E, you see in their synthesis pathway, you will not see cholesterol. In the synthesis of dolicol, it starts from acetyl coenzyme just like cholesterol, but after farnesyl pyrophosphate, cholesterol is formed after the precursor squalene, while farnesyl pyrophosphate directly gives rise to dolicol. So dolicol and cholesterol are not one after the other. Similarly, retinol is formed by carotene. This is we had already seen beta carotene, and vitamin E is again in this structure. You can see it's not from the cholesterol, right? Okay. This also you people had got right. So good job at that. Let's look at the next question. So which of the following are wholly or partly derived from isoprene? We already know isoprene. So which one of these are derived from isoprene? And the second question is very interesting. It says, order the following compounds with respect to the increasing oxidation state of carbon. So you know alcohol, take a basic alcohol and calculate the oxidation state of carbon on that alcohol. Similarly, alkane, similarly carboxylic acid, carbon dioxide, aldehydes. Now you have the oxidation state for each carbon. Place them in the order of increasing. So lowest to the highest. So let me know the answers. A, C, E, F, G. A, C, E, F, G. A, C, E, F, G. Okay. And yeah, the second one will be in order. So let me know the order of the yeah, options. A, B, C, E, F, G. Okay. 21534. Okay. I'll wait for the other person to also attempt the second question because it's easy, but once you do the exercise, then you're sure about it. Let's wait for a few moments for the other person to answer, and then we can discuss. So it's easy. Like you basically just write alcohols, alkanes, everything you jot it down, and then you just try to see what are the reduction state or oxidation state or carbon. So what is the oxidation state of carbon in that? And then you try to find out what would be the order. One more minute and then we'll discuss the answer. Second one, 2, then 1. Okay, and the other 3? Okay. Yeah, we can do it one by one. Just okay. So this is the answer. Okay. So the first one, I think all of you got that right. Right. So sorry. Just a second. Yeah. So the first one, you can see that we already looked into all of these compound ones or the others. We have dolicol, ubiquinone, vitamin A, D, E, K. These are fat soluble. These also compounds we have seen. Hello, am I audible? Am I still in the chat? I'm sorry. Oh, sorry. I thought I had missed it out. Yeah, just a second. Yeah. All right. So that was clear, right? Okay. Now next one. Sorry. The answer is 2, 1, 5, 3, 1. 2 and 1 you were able to do, right? You were able to do for 2 and 5. So similarly, you need to do for 5, 3, and 4. So if you do it for alkanes, I'm pretty sure you did the same thing. So the second one in the answer is alkane. So when you did for alkane, you found out that it was -3. Similarly, if you do for alcohol, you get -1. Similarly, you do for aldehyde, you will get +1. Carboxylic acid, you will get +3. And carbon dioxide, if you will get +4. Do you want to try it out once after knowing this? Maybe I'll stop for a few seconds here. Do you want to, with this information, do you want to calculate once again? Because you've already calculated for alkanes and alcohol. So I'm pretty sure you will be able to get for aldehydes, carboxylic acid, the same way you did for the other two. This is easy, right? If you look at CO2, yes, because if it's CO2, it's -4 here. So for it to be 0, it has to be +4 here. So it has +4. So similarly here, if you see 3, so 1, 1, 1, 3. So it has to have +3 -1, 1, 1. So -3 +3. That's how you are calculating for all the others. So +4, +3, -1, -3, and +1. And that's why the order is 2, 1, 5, 3, 4. Right? Okay. Next. So this is match the following. 8 seconds. Right. So you have lipids on your first column, types of lipids on your first column. You have names of lipids on the second group, which is again a classification. And the third one is a more specific example. So based on this table, can you give the correct base pairings or correct pairings in the answer? C. Okay. Okay. I'll wait. If that's the answer, then I will just discuss after 1 minute. Do you think there are other options to this?
M F G D B.
Sorry, which one? Sorry, I couldn't hear.
L F G D B.
FGDB. Okay, sure. So, do I have answers or are you planning to give one more answer, Aditi? Is C the final? Okay, all right. Maybe I'll give the answer first and then you can check if you were thinking in the correct direction. So, let me first give the answer C, D, E, and G. Okay, C, D, E, and G. Let us see first why. Let us just start. We'll look at the answer later. We'll just first see what's happening in the question itself. So, uh, let's first look at storage lipids. We have already seen multiple times what are the storage lipids. The storage lipid is triglycerols. So we, out of all these options, we'll know triacylglycerol is storage lipids. So one should always be C. Membrane lipids are glycolipids, phospholipids, and archibacterial lipids. This also we know from the classification. Sulfate groups are in glycolipids because we know there is something called what do you call sulfogalactolipids, which are in glycolipids. So this also is there. And phosphatidic acids are part of phospholipids. So we should have one C, 2A, B, D, 3A, and 4B. If we find any aberration in this order, we should discard that option right away. Why to complicate with the third option when we can just start by looking at the first two columns, right? So let's look at the first option. The first option says 1A. 1A is incorrect. We know one should be with C, so this one gets automatically cancelled, so not A. B, we know one is C, so we'll still keep it okay. Let's look at the third 2A, correct. 2D, correct. 2B, also correct. 3A, fine. And 4B, also fine. So we know the first step, we were able to discard this option, right? Now let's look at the next part, which is glycolipids. So now let's look at these options which are given. So we already know 1A is incorrect. Let's look at 1C3. So 1C, 3 is galactolipids, which is not a triacylglycerol, right? So we know that this is incorrect. 2A, 2. So membrane lipids, glycolipids, sphingolipids. This is possible. We have already seen this. glycolipids are glycolipids have the classification as sphingolipids. Then 2, D4 perfectly makes sense, right? So we got that right as well. 2B, phospholipids, can also have sphingolipids. This we know. We already saw sphingolipids can be in both glycolipids and phospholipids, right? Next, 3A and 2. Your glycolipids, sulfate groups should necessarily not be in sphingolipids. Sulfate groups can be in glycolipids, but they are galactolipid or sulfolipids. They are not sphingolipids. So that's why this is incorrect. And phosphatidic acid, phospholipid, which is glycerophospholipids, as we have already seen. Let me know if you have any doubt. If you have any doubt, please let me know. We can again go back to this slide and see. Clear? Yeah, we have already done this classification. That's why I said that that classification chart is the most important. You remember that your lipids are very easy to remember. Right. And this is probably the last question of lipid section. So yeah, which are the following are the functions of dolichols? And there are some questions that you don't, that are of course not correct. So I've also given the correct answer for them. But let's first look into the answers which you give. So which are the following are functions of dolichols? They are involved in the assembly of complex carbohydrate, assembly of peptidoglycan. They are lipophilic electron carrier in ETC, electron transport chain. They function as antioxidant, hormone. They are regulation. They're important for regulation of calcium uptake in the intestine, and they're involved in carbohydrate units to addition of carbohydrates to leucine residues. A, B, and G. Okay, just a second. All right, A, B, and A, B are correct. And that makes sense. Dolichols are part of complex carbohydrates and they're in the assembly of peptidoglycan. While this C, which says that dolichol is lipophilic electron carrier, that's incorrect. But that one is ubiquinone. Okay. What functions as an antioxidant? Where generally vitamin E, vitamin C, and ubiquinone function as antioxidant. Dolichols don't do that. Hormone, we already know there are so many hormones, and we never read about dolichols there. The dolichol is not a hormone. For calcium uptake, I am pretty sure we all know that vitamin D helps you in uptake of calcium. So that's regulation of calcium is for vitamin D. And lastly, dolichol uh, do add carbohydrate, but that they do on asparagine and not leucine. So the wrong part here is the residue. The residue is not leucine, but the residue is asparagine. But rest you got correct. So great job there. All right. So now let's move to the next part of this session, which is also the second part of this week, which was about metabolism, where we quickly touched metabolism. Remember, we'll do metabolism in more depth in the next session, where we'll look into bioenergetics and the aspects and principles of it. Today, we'll only look into the basics of metabolism and the type of reaction that are involved in all the metabolism pathways that you see, right? And today's class will be short because we'll only have this topic to cover and I think one or two questions on them. But next session we'll have more on bioenergetics and glycolysis pathway, and we'll try to work it out properly for all the number of ATP molecules and everything else. All right, let's come back to this. So second part of our session today talks about metabolism. So a metabolic pathway is basically a set of interconnected chemical reaction that occurs within a cell. The reactants, the product, and the intermediates of an enzymatic reactions are referred to as metabolites. So I will give you an example from the example that we have learned today. All right, this is a pathway. So you start with acetate, you have cholesterol, you have pregnenolone, you have all these different component. These are all metabolites. All these intermediate components are called metabolites. And this is what we were talking about in this slide. So all these reactants, all the products, all the intermediates, they're all called metabolites. So you can have one pathway, two pathway, and three pathway interconnected. How is that? So let's say the first pathway has six steps or five steps, which starts with reactant A and production of F component. So you have A to B, B to C, C to D, D to E. These are all enzymatic steps. And remember from the previous discussions on Michaelis-Menten's equation, any of these, whichever is the rate limiting step, determines your Michaelis-Menten equation and how does the enzyme kinetic looks, right? But what we are more concerned here is, let's say A converts to B, B converts to C. The final product is F. So whatever forms in between, they are called intermediate. And that's not a what do you call why should we have these intermediate, why not directly from reaction to product? Well, the intermediates serve their own purpose. And what are those purpose? They are each a precursor to something else in the another pathway. So a C converts to D in pathway 1, but that same C is converted to G in pathway 2, which gives rise to H and I. So the C from the first pathway is required to give the product I. if the, if this pathway is affected, not only F is affected, I is also affected, and by the same logic, K is also affected. So regulation of a pathway is very important. uh, we'll do when we do glycolysis, we'll see why we have regulation, why we have allostatic regulation, why we have reverse pathways acting into this like into the pathways, because we regulate that concentration so that our product and there is a homeostasis is maintained. So you will see C coming to the second pathway giving rise to I, but the same pathway second intermediate G gives rise to something other product which is K. So all these products, all these reactants, all these intermediates are metabolized, and all these chemical reactions together will give rise to something called a metabolic pathway, because they are all part of pathway and they are all involved in some or the other kind of metabolism, right? Now, metabolism by itself has two processes, catabolism and anabolism. What is catabolism? When you have larger molecules for cytoplasmic membrane, cell wall, or ribosomes, and when you break them into smaller molecules, which are like amino acids or basic components, which are called, this is called the catabolism. Okay, when this happens, you have exergonic reactions, so you have heat generation, and you also produce ATP. When you are breaking something, you are utilizing energy in currency in the form of ATP, using that energy, giving out the heat and breaking the bigger molecules into smaller molecules. We know that this kind of reaction or this kind of pathways take place during glucose breakdown or glycogen breakdown. So all the breakdowns of bigger molecules is by catabolism. Catabolism is basically breaking. The reverse is smaller to bigger, which is anabolism. So the smaller components, when they are stored, they are basically all the smaller molecules are all polymerized, or are they all like they're formed one single chain. So that's larger molecules. So both of these reaction take place, and that together constitute the metabolism. You have catabolism and you have anabolism, right? So there is simultaneous synthesis, which is, let's say, here, and simultaneous degradation, which is here. So this is synthesis, larger molecule is being synthesized, larger molecule is being degraded. So if you have simultaneous synthesis and degradation, that would be wasteful. And to prevent that, it is so to prevent this kind of wastefulness. Why is this wasteful? Because then you don't have one single component in the cell at all. Let's consider this from a cell perspective. So glucose together is stored as glycogen. Glycogen is broken up to give rise to glucose component. Unless we take glucose from outside, let's just keep that for a side. So you have in your cell, glucose content has gone down. You want glycogen to break down. So your glycogen breaks and gives you new glucogen. But if you have simultaneous anabolism, the same glucose will again polymerize and form glycogen. What's the point? That's no usefulness. You broke the glycogen down because you need more glucose. But if you don't retain it at that step itself, then it again goes back and forms the glycogen. It's basically no use. So that's why simultaneous synthesis and degradation is a wasteful way. And this is prevented by reciprocally regulating the anabolic and catabolic reaction. And that, how is that determined? That is determined by equilibrium. It is determined by concentration. If let's say too much of a concentration inhibits the formation, so you have a lot of glycogen already in your, a lot of glucose in your blood already. If you have a lot of glucose, you don't need any more glucose, so that catabolism will be stopped. Similarly, if you have a lot of glucose, there is no catabolic reaction, but still you have a lot of glucose. The glucose are then start to combine, so they are taken up and they're stored as glycogen. So determining what is the concentration, where are, which cell are these components present, that determines the which of the reactions will take place, catabolic or anabolic, not both of them simultaneously, right? And because it is, they are regulated separately, a catabolism or a catabolic pathway is necessarily not like antagonistic of anabolism. So anabolism is not identical to catabolism in reverse order. Why? What does it mean? So when I said glucose to glycogen, that pathway, and glycogen to glucose pathway, when you look at the final product, they are similar. Yes, glucose to glycogen, glycogen to glucose, they are opposite in nature. So you would expect that these intermediates would also be going in the reverse. That's not correct, because at least one of the steps in these two pathways, they're catalyzed by different enzymes. So the moment you have different enzyme, you have different regulation coming into play. And because you have different enzyme, they are regulated by different kind of substrate or reactant. So your regulation pathway differs. So that's why the pathways, even though they have the same reactant and final product in the reverse order, they are necessarily not reversible in nature or reversibly identical in nature. This is very important, and you need to remember this. Catabolism and anabolism does not mean that they are identically opposite. Okay. Now, along with this, these anabolic and catabolic pathways are also irreversible, because one is that one of the steps is different enzymes, so they are not identical. Second is they are irreversible because one of the steps here is thermodynamically very favorable. So thermodynamically favorable means that that intermediate will form and it will not go back in the opposite direction. So they are irreversible in broader sense. Essentially, they are irreversible because when you see the steps of different pathways, one of the step is thermodynamically extremely favorable, and that means that it's very stable. So the moment the product forms or the intermediate forms, that's held there at that step, right? Okay. So now here, this is what a basic gist of this is what we saw in the previous slide. So you have in a metabolic pathway, it's not linear, and it's basically non-linear. Non-linear means it's not straight pathways that okay, product. Linear means this simple straight line, but non-linear means this moving to this moving to this. You can also have back where it can go back to this interaction. It might even help form the precursor to A. So it's non-linear essentially. So this non-linear pathway, they basically have three main classification. So they are basically either converging catabolism, diverging anabolism, or cyclic pathway. Then this, if you know, this is acetyl coenzyme A, which is going to Krebs cycle, which is forming oxaloacetate acetate. Before this, you have the glycolysis. From this, you have several other formation. All of these pathways, you would definitely study in biochemistry, and you will see when you are done studying all of these individually, you will see that all of them are connected somehow or the other. And that's because metabolic pathways are not linear. So you will see that, let's say this is the breakdown of fatty acids. So you have TAG for, or this is the formation of fatty acid. So you have 3AG to THG breaking down to fatty acids, phospholipid breaking into fatty acids, starch, glycogen, sucrose breaking to glucose or alanine, serine, all of them forming acetyl coenzyme A. So all of these are converging to acetyl coenzyme A. They're all catabolism reaction because they're all breaking down, and they're one of their product will be acetyl coenzyme A, either the final product, or they are converging, or they're going towards the formation of acetyl coenzyme A. Now this acetyl coenzyme A is an anabolism diverging. So this acetyl coenzyme A is a intermediate for so many reactions. You have acetyl coenzyme A. If it goes to fatty acid, if it gets like bond to fatty acid, it gives like to several different kind of derivatives. You can give icosanoids, triacylglycerol gets attached to diacylglycerol, gives you phospholipids. This is all about lipids. If it goes to mevalonic acid pathway, it forms vitamin K, it forms cholesterol, it forms rubber, carotenoid. These are all secondary metabolites, steroids, bile acids, all of them, right? This is diverging and this is converging. This is converging because they all of these pathways are converging on one single component. Diverging because one single component is giving you different kind of products, right? And it can also be cyclic. This same acetyl coenzyme A can be incorporated when oxaloacetate converts to citrate. That citrate converts to different other intermediates and comes back to oxaloacetate. And then again, it is recycled. So here in one of these pathways, acetyl coenzyme A is given out. That acetyl coenzyme is again used and again made use here, returned here, made use here. So it's cyclic. So either you have a cyclic pathway, or you have a diverging anabolism, or you have a converging catabolism. Converging catabolism, why? Because you need that one product which is a unit product. So a smaller compound can come by catabolism, so you converge on that. And diverge because a smaller compound, it can get attached to other components and then give rise to a bigger component. So diverging anabolism. So all these are different kind of pathways that you find in metabolic pathways, which of course are non-linear in nature. Now, this is the last topic for today, and then we can wrap up. So, in any metabolic pathway so far that we have seen, let's say glucose to, so here let's say glycogen to glucose, triacylglycerol to fatty acid, the 5 reactions that we will see now, one of the reaction is uh, in the steps that you will see. Now, what is the most basic kind of reaction? That's oxidation reduction reaction. Redox reaction. Something is getting oxidized, something is getting reduced. Easy to understand. If something is getting oxidized, it means it's losing its hydrogen, and something is accepting that hydrogen and electron, it's getting reduced. So you have alkane, alcohol, aldehyde, carboxylic, carbon dioxide. These groups, they will take part in redox reaction. So you can see this when we will see glycolysis, we will see lactate is formed, and we will see lactate gets converted to pyruvate. This is via a redox reaction. So you have lactate has a CHOH group that gets oxidized, and we will see there is a redox reaction taking place. So one of the main reaction classes is redox reaction. The other is carbon-carbon bond formation. So let's say this one. So here you have a CO group and you have a C here. So because of these electron transfer, you will see a double bond formation between C and C. So C and C get double bonded. And what kind of reaction use this kind of double bonds? Aldol condensation, Claisen ester condensation, and decarboxylation of beta keto acid. So what happens here? Let's look at aldol condensation. So you have a group here with CO and R2 and H, and you have another group which is again a CO and R3 and R4. Now electrons from here will attack these will get bonded such that you have a CC bond formation, and you have these two components are joined by the C and C. So the carbon of the two groups get connected. So this is aldol condensation. Okay, here this group here got converted to aldol. So alcohol is so H is added there. This electrons are taken and transferred to this H finally. Okay. Or you can have this Claisen-Ester condensation where you have the same compound, same kind of group, but what is happening instead here you have the R1 and R2 groups. So there is only the other component has R group, so you are adding that this Claisen-Ester formation here. And here you have decarboxylation. Basically, you are removing these NCO minus as CO2. So your CC bond has broken. It's not always the joining, it can also be breaking. So you have a carbon-carbon bond formation or breakage, whichever. Third is generally group transfer reaction. So let's say you have this group, this is the compound, and you have a Y group. You are transferring Y onto the original compound, so you will remove one of the extra group. So what happens, you have this intermediate tetrahedral, which is highly unstable. So what happens is due to electron transfer, your H groups compound and Y groups attached. And you will see a lot of these reactions. So let's say here, you have adenine, ribose, and the ATP and a glucose, which is to be added. So the glucose gets added as in phosphoester bond. So you get connected, and this is the kind of group transfer because this is getting connected. This is not via CC bond. So this group is getting added. So it's a group transfer. So you have CC bonds, you have group transfer, you have redox reaction, and next you have free radical reaction, which are very easy. So free radicals is basically, uh, let's say there's a bond between A and B. This gets broken because of heat or light. So the energy gets both of them as free radical, very charged, and then they propagate that charge. So let's say A now goes to C and D, breaks a C and D bond, forms a bond with C. D forms as a free radical. Then you have D, which is a free radical. It can oxidize, so it breaks this bond, it forms. So this way it is going. When does this finally finish or terminate? When you have these same kind two free radical coming together to form a bond. So you have free radical propagation. So that is also, and you will find free radical especially in peroxidase. So your peroxisomes, you will find a lot of free radical reaction taking place, right? And the last category is isomerization or elimination reactions. So isomerization is basically one form of compound gets isomerized due to transfer of one group from one place to another, and it forms a second group. So you have glucose 6 phosphate. You can look at this. One, two. First one has the double bonded O, and the second one has CHOH. Isomerase, phosphohexose isomerase. It is phospho because it's phosphate group. It is hexose because it's glucose. So glucose has 6 carbon. So phosphohexose isomerase, what it does is it just takes this O to this second group and transfers this HOH to the first. And that makes it a fructose 6 phosphate. And we have looked into glucose 6 phosphate and fructose 6 phosphate in the previous session. We know the structure. They are structurally different at the O group. And we can say an isomerase can convert this from one compound to the other compound just by isomerizing the component, right? And you can see how that actually takes place. You have these reaction here. So let's say you have CO and COH in this bond, which is exactly what we are doing. And what it makes use of is a proton from another compound. So that proton is taken, and that because there is a proton here, this attacks this, so this makes you formation of this C double bond C, and then electrons from this uh, carbonyl group form an OH bond with the hydrogen from B2. So you have different groups on the other side also. Makes use of the protons from both the sides, and this gives you an intermediate which is highly unstable. Finally, it breaks down to transfer the OH group from the second to the first and O from the first to the second, and that gives rise to the isomerization. Similarly, here if you see, you have CHHR and the second one has CHR1 and OH. Isomerization leads you in a trans configuration RR and H. So the H2O is removed. So you can remove water, eliminate it, and it will give you the isomer or a different configuration or conformation of the product. So you have either isomerization or you have elimination of water, and that gives you a different component. So you have these 5 main classes that take place in metabolic pathways. All of these are formed in or made use on different pathways, and we have different components and different pathways that converge on each other, right? And we have two questions that will sum up today's lecture, and then that's it for the day. So let's do the first question, which is, so you have different, we have already discussed this right now. So it shouldn't be very difficult. You have these three different types of kind of reactions, and the three different kind of reaction on the second column. You need to match and tell me which one is correct. So you can look at the uh, reaction and guess what's happening. So you can see one CO2 is getting removed. Here you have CNC getting formed, and here you have yeah, you guess here what's exactly happening. So your CNC are forming, but your this group is getting removed. So what exactly is happening? We have all discussed right now. Yeah, C1, okay, C1, okay. A2, okay, V1. Okay. So C1, A2, B1. Are you sure? You've repeated B3. Okay. C1. Okay, I can see someone else answer. Wait, I'll wait for the answer. Okay. C1. Uh, I'll wait for a few more seconds for you to answer the other, and then we'll look into the answers. Okay. Uh, P2. Okay, you're changing your answer. A3. Sure. I'll look into the answers. Okay, B2, A3, C1. Okay, let me see. You, sorry. B2, C1, A3. And that's the answer. That's exactly what is happening. I hope you got the answers in what sense we are saying. So we are saying that C is for 1, B is for 2, and A is for 3. I guess you wrote the same thing. Yeah, right. So as you would have already guessed it, so the reaction 1 here, you can see it shows decarboxylation very clearly. You are losing a C here. And we have already seen this. This is a beta keto acid decarboxylation, right? Reaction 2, as you see, it says it is showing the formation of a beta hydroxy compound. And we have seen CC formation happens in aldol condensation. We have already seen this, right? And the last one here you are removing this HSR2. So this is where an enolate attacks another ester. So this enolate attacks another ester, and this forms a beta keto ester, thioester. So this is Claisen ester condensation because this is ester condensation here. So you are able to get to it finally. And we have already looked into all of these in the previous slide. So here we already looked into all of these. So this is your, just a second. So this is your Claisen ender. This is what exactly we saw right now. This is the carboxylation, and this is aldol condensation. So you were able to get it right. So good job there. And the last for the day. So you have 4 statements. Let me know which one is incorrect with respect to metabolism. So both anabolism and metabolism are not identically reversed. At least one of them is catalyzed different ways, true or false. Both anabolism and catabolism are irreversible as at least one step is thermodynamically unfavorable in reverse direction. Anabolic catabolic pathways often operate in the same subcellular components, and most biochemical reactions are dominated by the following principle: breaking of covalent bond and electrophile nucleophile interaction. This I will come to, but first let me know our answers to this question. C. Okay, that's it. That's your answer. 10 more seconds and then we can discuss. Okay, that's it. All right. Give me 10 more seconds so that if someone else is attempting, I'll let them answer. B. Okay. All right. So I think I've got the answers to it. Just a second. So the answer which is incorrect is C, right? And we'll see how. So, okay. So we have already done the anabolism, catabolism reactions, and we have seen. So let's look at the first one. First one says anabolism and catabolism are not identically reversed. At least one step is catalyzed by a different enzyme. This we know is true, right? Because we said that we will see, then when you see, we've already taken the example, glycogen to glucose and glucose to glycogen. If you see them in the reverse order, let's say you see formation of glucose to glycogen, there are a set of enzymes, and the breakdown of glycogen to glucose, a set of enzyme, you will see they are not exactly identical. That means not each step is just simply reversed. There are some steps which are done with different enzyme, and that makes it being regulated by different components, and that's how they are regulated. So this one is correct. They are definitely not identically reversed, and because they have different enzyme. Second, both anabolism and catabolism are again irreversible, as at least one step is thermodynamically unfavorable in reverse direction. And we had already seen this. This was one of our points. That not only is it regulated by different enzyme, one of the steps in either this formation or in this breakdown is enzymatically favored in the forward direction, such that it is very stable and it does not go back. And that's how it regulates that step, that pathway. So one of the steps is irreversible because of thermodynamics. Because thermodynamically it is favoring the forward reaction. And because once the forward reaction happens and that intermediate product is formed, this does not go back. For you to go back, you need to reroute and take alternative enzymes. And that's how it's regulated. That's how it ensures that not everything is going back identically. So one of the step is either by different enzyme, or one of the step is highly thermodynamically favorable such that it does not go back in the uh, reverse direction. Okay, we will see more of this the moment we do glycolysis in the next session. We'll not only do glycolysis, we'll also look into the regulation of glycolysis. That will make this more clear. And fourth is also correct, which is most biochemical reactions are dominated by following principles, which is breaking of covalent bonds and electrophile nucleophile interaction. So all the bonds formation that you see, they may be different types of reactions, but the inherent feature is forming of covalent bond and an electrophile and nucleophile attacking each other. I will call it covalent formation or breaking or nucleophile and electrophile attacking each other. That's exactly what you saw in the previous slide as well, right? You have this charge components forming, or you have a covalent bond formation. So all of them follow these two main classes of reaction. Now, what is incorrect? Incorrect is anabolic and catabolic pathways operating in the same subcellular components. Remember, we said that they are, it's wasteful to have them happen simultaneously, because what did I say? That why are we breaking glycogen? Because we have less of glucose in the cell, and we are not uh, like there is no uptake of glucose or intake of glucose. So how does the cell maintain the concentration of glucose? By breaking down glycogen. Now, breaking down of glycogen gives you glucose molecules. If you uh, if there's a simultaneous synthesis and degradation, this glucose form will again polymerize and form glycogen. What's the point? The cell is still not getting new glucose. So you don't have it in the same compartment, because that's the way of ensuring that there is no wastage of product. So they are generally compartmentalized. If your anabolism or anabolic pathways is taking in one set of cellular compartment, your catabolism takes place in other set of cellular compartment. That's to ensure that none of the products are used wasted. And if there's a crosstalk, that crosstalk is via the intermediates. This intermediates comes out and forms the as a checkpoint. We'll have all of these in glycolysis or in whatever pathways we see: checkpoints, regulation, concentration, so all these will ensure that the pathways maintain the hemostasis, maintain the correct concentration of the components in the cells. Now, any of the enzymes dysfunctions or gets improperly formed, you have a lot of problems downstream, because we have already seen, first of all, there is a lot of crosstalk. One intermediate is not just one for one pathway, it is part of many pathways. So if you have one enzyme which is not functioning, the product doesn't form and everything else falls down. So that's why everything is properly regulated. And that one part, or the important tenet of that regulation is compartmentalization. So your anabolic and catabolic pathways are not in the same subcellular components. They are generally compartmentalized, and they are in different steps, in different ways. So we will see that fatty acid synthesis generally takes place in cytoplasm, while the breakdown, which is oxidation, takes place in mitochondria. So your breakdown is in mitochondria, and your formation is in this thing, cytoplasm. So you remember, right? Mitochondria is a different organelle altogether. Cytoplasm is the everything up outside, everything apart from the nucleus in the cell. So you have this kind of compartmentalization for all the anabolic and catabolic pathways. And that's why this was a wrong answer. Okay, and that brings me to the end of today's session. As I had already mentioned, that this was a more of a theoretical session, so we had no numericals. And if you want any numerical, any topic to be discussed, please put me a mail or put it in the discussion forum, and I will try to incorporate that. And next session, as I have mentioned, we'll do glycolysis. We'll have bioenergetics and glycolysis, and we'll do glycolysis very nicely so that we understand every step of it and what do you finally get out of it. So how many ATPs, how many NADH, how many FADH, and how do they convert to the total number of ATPs that are generated. So we'll do all of these in the next session, and that's it for today. If you have any doubt about any of these topics and you want to be discussed, let me know. Otherwise, we see each other in the next week.